Welcome to this poster presentation. ISPRED-SEQ is a novel deep learning-based tool for the localization of putative interaction sites on a protein sequence. The method was developed by members of the Bologna Biocomputing Group, under the Department of Pharmacy and Biotechnology of the University of Bologna. Knowing how proteins interact in the cell context is critical to fully understanding the functions they perform. However, this task can be addressed at different levels of resolution. On a lower level, we want to identify pairs of possible interacting partners. This process allows the generation of huge interaction networks. On a higher resolution, we want to fully characterize the interacting surface for each pair of known partners. A third approach is to map all residues of a protein that are known to be involved in different interactions, regardless of the specific partner. These residues are defined as protein-protein interaction sites, and their identification is the goal of ISPRED-SEQ. After performing the annotation on the structure, it is possible to map it back on the protein sequence. In this way, our method is trained without ever observing three-dimensional features, making it able to generate predictions for proteins that are lacking an experimentally annotated structure, and thus greatly extending its applicability to large-scale studies. ISPRED-SEQ is published in the Journal of Molecular Biology. If you want to read the full paper, please scan the QR code you can find on this slide. The main advantage of ISPRED-SEQ over other methods performing similar tasks is the adoption of embeddings coming from protein language models to encode the target sequences. Protein language models are deep models trained on millions of protein sequences, with the task of reconstructing as output the proteins they observe as input. This seemingly trivial task actually puts a strong constraint on the internal layers of the model to capture as much meaning and context as possible, effectively learning to understand the language of the proteins. After discarding the output layer, the model that generates the internal representations is made available to the scientific community, and it can be employed to embed new sequences for any kind of downstream predictive tasks. Training such huge models can require even months of computation on high-performance TPU and GPU. Nonetheless, encoding a new sequence takes only seconds even on personal computers, making it a one-time investment that quickly pays off when compared to the cost of computing multiple sequence alignments. This QR code can point you to a few key publications on this topic. After an extensive grid search with a stringent cross-validation procedure, the final model architecture of ISPRED-SEQ adopts two different and complementary protein language models to encode the input, ESM1B and PROT-T5. Each residue is processed independently by the network, after considering a window context of 31 neighboring residues. Each input matrix is first processed by a convolutional layer that captures local patterns in the window, and then by a stack of dense layers to generate a binary classification, distinguishing putative interaction sites from other residues. ISPRED-SEQ is freely available to the scientific community in the form of a web server, where users can upload a target sequence and visualize the results. Scan the QR code if you would like to try it out. ISPRED-SEQ was trained on more than 6,000 non-redundant protein sequences and benchmarked against state-of-the-art methods on a blind test set called DSET448, which is routinely employed in the literature to evaluate the overall ability to perform this task. It can be observed that both datasets have a naturally strong imbalance in the number of positive and negative examples, making the problem much harder to tackle. Nonetheless, thanks to the adoption of protein language models, ISPRED-SEQ greatly surpasses the performances of all other methods included in the comparison. Most notably, when focusing on Matthew's correlation coefficient values, we obtain a 7 percentage points increase over the second best performing tool. Moreover, the adoption of protein language models in place of canonical representations based on multiple sequence alignments makes ISPRED-SEQ extremely fast. Thank you for your attention, and see you in the on.